Okay, we're very excited today to have Matt Bochenek here. He's coming to us from MIT. The talk title that he's giving today is Wireless Oxygen Generating Device for Long-Term Cell Therapy in Type 1 Diabetes. And a little bit about him, he's a postdoctoral fellow in the Anderson and Langer labs at MIT, where he develops immune-protected cell therapies for the delivery of protein biologics. His research focuses on overcoming key barriers to translating encapsulated cell technologies from bench to bedside, including the design of immunomodulatory biomaterials that prevent fibrosis and the engineering of cell-based systems to remain viable and functional even under the hypoxic conditions typical of implanted devices. Dr. Bochenek is particularly interested in enabling cells to act as smart, stimulus-responsive drug factories, for example, producing insulin in response to glucose as a therapy for type 1 diabetes. And his work bridges material science, immunology, and cell engineering with a strong emphasis on scaling from rodent models to large animals, including non-human primates. These efforts address critical challenges in clinical translation, such as identifying optimal transplant sites in bipedal animals and navigating the complexity of the primate immune system. So ultimately, his goal is to develop next generation cell-based devices that can safely and effectively deliver therapeutic proteins in humans without the need for systemic immunosuppression. I will drop his latest paper into the chat, and we're very excited to have you. Thank you very much for joining us.
Yeah, thank you very much, Monica, for that introduction. Maybe I shouldn't have given you such an extensive background but you did very well. And yeah, thank you very much, Sugar Science, for having me. I'm excited to kind of present some of the work we've done on developing this wireless oxygen generating device for islet transplantation as a potential therapy for type 1 diabetes. And so all
Right.
this work was done in collaboration with Sid Krishnan while he was a postdoc at MIT, and he's now an assistant professor at Stanford. He's a really talented engineer in bioelectronics, material science. So it was, really a privilege to be able to work with him.
Can I just ask, is he still involved in this type of project?
So he is, but he's also kind of looking at it from, he's working on this in his own lab, so trying to kind of come up with kind of enhanced designs that maybe can, help some of the potential challenges or limitations that we run into, kind of
Great.
on the current kind of the device design. Um,
Nice.
so yeah, we're still collaborating. I think I'm meeting with him on Tuesday, which will be nice. Um, so yeah, in general, I've been kind of working on immunoisolation devices for cell therapies. So the general concept is we want to protect therapeutic cells within a semi-permeable membrane or barrier where the cells can receive kind of nutrients they need to survive or produce therapeutic proteins like oxygen, glucose. The secreted protein of interest can diffuse out of the device to regulate the disease. However, they're protected from the immune system by this porous membrane that prevents immune cells from getting within the device that would otherwise destroy the device, destroy the therapeutic cells. And so we're trying to create this living drug factory where the cells will continue to produce the protein drug of choice without kind of needing to repeat injections or doses or require immunosuppressive drugs. And so these devices can use primary cells like islets that are isolated from human donor organs. We can use genetically engineered cell lines that can secrete therapeutic proteins like say like erythropoietin, EPO, or even biologics and some of these like antibody-based therapeutics. And then lastly, we can use differentiated stem cells, so like SC islets, for instance. And there's been some really exciting work just recently published by Vertex, kind of using those cells, but intraportally transplanted and under immune suppression. So hoping that we, through these devices, then we won't have to, give patients immune suppression, which can limit the types of patients like that can, are eligible to receive these, that type of therapy. Um, And so I don't want to go into too much detail, but type 1 diabetes results from an autoimmune destruction of insulin producing beta cells. The current kind of standard of care is blood glucose monitoring. It can be manual insulin injections, or even now there's continuous glucose monitors and insulin pumps. But it's really been difficult to fully recapitulate the dynamic glycemic control that's seen in the body. So islets are really, complex cell clusters that, with different cell types that secrete different hormones to really maintain that tight, control of blood glucose levels. And so once you have, say, imperfect blood glucose, blood sugar control, that can lead to long-term complications, increased morbidity, and really just like an overall burden on the lifestyle of patients and their families. So really, so you can see kind of from this is kind of representative of a continuous glucose monitor of a patient with type 1 diabetes using manual insulin injections. And so you can see, they're, outside that kind of window of where we want to be in terms of level pretty, often throughout the day. And like even when we use the most advanced kind of like insulin pumps and with the CGM, like we're still kind of outside that that window, and that can kind of lead to those higher A1C and those long-term complications. But if you look at a non patient without diabetes, their glucose levels are really regulated, really tightly regulated. Um, and so that's what we're trying to accomplish. Can we replace kind of the beta cells so that they can restore the physiological kind of cellular control system that has evolved to really tightly regulate and maintain blood glucose homeostasis? And so hypoxia has been a major challenge for encapsulated islet technologies. And so, islets vary in size from really small, 50 micron, all the way up to kind of 300 micron in diameter. But the cells in the core of the islet, since they're not revascularized anymore, you have a lot of central necrosis and a lot of cell death, just because those cells in the core don't get any, enough oxygen. And so even if you kind of spatially pattern them, you can still see that, right, those oxygen tensions for those even small clusters at high densities, are in these kind of hypoxic states. And then this is just exacerbated when we look at minimally invasive transplant sites like the subcutaneous site, which is kind of a holy grail, right? Just placing, say, like a device or a transplant under the skin, would be very easy for patients, and a surgical, not a very extensive kind of surgery. But this, transplant site has low partial oxygen pressure that makes it even more difficult to protect and get these cells to function. And so insulin secretion by the beta cells is heavily dependent on the availability of oxygen. And so this graph just shows the fraction of insulin secretion by beta cells under different hypoxic conditions compared to normoxia. So, if you basically reduce like the oxygen to say like around like 5%, they're secreting roughly almost like 10% of what they would in kind of a normoxic and situation. So with our device, we are trying to, supplement oxygen to provide that essential kind of nutrient that's required for insulin secretion. So here's just the overall kind of cutaway of the side view of the device. So we put the islet cells within this cell chamber, and then they can, secrete the therapeutic protein, insulin in this case. And then they're protected from the immune system by this nanoporous immune isolating membrane. So it's a polycarbonate membrane that's produced off the shelf. And then inside, we use a proton exchange membrane, which uses vapor-based electrolysis of water inside the body. So it'll split, if you apply a current, it'll basically split water in your body into oxygen gas, which will feed the cells, and then hydrogen gas. And so hydrogen gas will then kind of diffuse out the back of the device. And this is completely implantable, so it uses wireless power harvesting to power the device through the skin. And then, all these components can combine into a quarter-sized implant. So, this is really important when we're, want a really small device, which would be kind of the most, well adopted by patients. And so here's kind of a diagram of what the membrane electrolyzer looks like. So we take that proton exchange membrane, we sandwich it between kind of two electrodes. And then once we apply a current through those electrodes, then that will split water into oxygen gas and hydrogen gas. And this is, really linearly correlated to the current that we apply is how much oxygen kind of you're generating. And so we can really optimize and kind of dose the amount of oxygen specifically to say what islet source we're looking at, kind of the density of cells that we have within the device. And so it's a really, really nice system. So now kind of looking at a broad overview kind of the device, kind of some additional components. So we have a support frame, which really kind of creates this like kind of mechanical kind of compression that we found is really important to have these kind of implanted electronics work long term. Then after we basically, this generates the oxygen. So then this gets transferred into this gas diffusion chamber, which holds the oxygen. And then this is separated from the cell housing chamber by a very thin kind of PDMS layer, about like 100 micron. And PDMS is permeable to gases, so to oxygen. So then the oxygen will slowly diffuse to support the transplanted cells. And so this is kind of what the device looks like while it's functioning. So you can see the bubbles kind of being created while it's being powered. And then we can also kind of tune the amount of oxygen that we need by kind of cycling or doing different pulse modes. So kind of like turning off on and off. We can really maintain kind of whatever kind of desired partial oxygen pressure that we want. So kind of now that we have the design, so kind of the first experiment that we did is how will this kind of support islet cells? And we used a hypoxic incubator chamber where we could incubate the cells at either 1% or 5% oxygen, culture them for 4 to 7 days, and then kind of look at how we're able to affect their viability and their glucose responsiveness after being in these kind of low hypoxic environments. So you can see islets at kind of 1% oxygen without any supplemental oxygen. We have a lot of cell death, really poor viability. However, once we provide kind of supplemental oxygen through the device and water splitting, we can maintain really high viability that is comparable to incubating islets at a 21% incubator. So then another kind of important measure, we need viability, but we also want to make sure that the cells are glucose responsive and secreting more insulin, say under high glucose compared to low glucose. So when you look at, say, kind of encapsulated or non-encapsulated islets at 1%, they really have lost that ability to secrete insulin in response to high glucose. However, when we supplement them with oxygen, even in that 1% kind of environment, we're able to maintain that really high responsiveness and insulin secretion, which is really like necessary for ideal function of these cells. So then another kind of reason why we are basically supporting the cells by giving them supplemental oxygen is that this allows us to hopefully increase the density of cells that we can pack within the device, so we can start to approach device sizes that would be adopted by patients. So you can look at some of these kind of earlier, even more recent from like our lab. These devices, they're approaching about like 600, 700 centimeters squared. So that's, that's like the norm. So it's kind of like putting almost like a TV like on your back. So it's, it's not really feasible by a patient. So they're not really gonna, want to get a transplant like that. So you really need to kind of increase this density to around 5,000 IQ per centimeter squared, and then you can get a device that's maybe a credit card size, maybe like a little bit larger, maybe about like half the size of a iPhone, and that could be that might be adopted by patients at that small kind of size. So the first experiment we did kind of went back to that hypoxic incubator, incubated the cells at 5%, so just kind of like a little bit higher and representative of the subcutaneous transplant space. And so we don't really see too much of a difference between in terms of viability for the cells after for day 7 when we supply them with oxygen or just in that kind of environment. However, when we look at their glucose responsiveness, only when we support them with additional oxygen are we able to kind of maintain that high stimulation index or increased secretion of insulin under high glucose compared to low glucose. So this is really important that we can kind of achieve these high density of cells within the device. And so kind of the wireless power transfer, how it works is it all works on kind of resonant inductive coupling. So kind of similar to your, say, like your iPhone, putting your iPhone on a charging pad. That's kind of how it's harvesting that power. So this is what we use for in vitro studies. So we have this kind of pad that has our transmitter antenna, and then we have the receiver antenna is on the device, which then kind of harvests the power to generate oxygen. So that's what we use for the in vitro studies. But right, we want to test to see how this will work in vivo setting in an animal. And so for this, we used a Neurolux cage. So here, the cage actually acts as the transmitter antenna. So by kind of wrapping these coils around the cage, we can create this like NFC zone where the animal can move around freely and it's continuously harvesting kind of power within the cage. And so this is just kind of a schematic of kind of some of the details of how it works. And then kind of prior to the first kind of implant, we measured, how much current is being produced, like how much power are these devices, harvesting within the cage. So we kind of measured this within kind of different spots in the cage just to make sure that we were getting kind of a uniform power harvesting across the cage. So then for the first experiment, we took donor rat islets and then put them about 850, loaded them within the device, and then transplanted into STZ diabetic black 6 mouse. And so first we followed these for 1 month so we can see nice glycemic control by our oxygen macro devices, whereas the if we don't supply kind of additional oxygen, we only get to about 300 mg per deciliter, and then kind of quickly returns to a hyperglycemic state. And then kind of another important thing is we want to kind of remove the device and then see that the animals return to a hyperglycemic state. So that kind of proves that the glycemic control was due to kind of the function of the device. And so here's just kind of an AUC. We don't see a significant difference between the naive non-diabetic animal and ones transplanted with our oxygen macro device. And it's like really well tolerated by the animals. So here is kind of after 30 days, we see that the fur has grown back, kind of, and it's really well tolerated, kind of by the mice. So this was all really, really nice that we could achieve kind of 1 month glycemic control, but we really want to extend this to, at least like 3 months, 6 months, maybe even up to a year. And so we kind of improved on this device through iterations to kind of improve the function. And so for these experiments, we used a genetically engineered hex cell line that secretes EPO, which is as a kind of a model therapeutic protein, and then kind of the same subcutaneous device transplant. And here, so the Gen 3 was statistically significant, compared to say our sham and without any oxygen. But now with this Gen 4, we're really getting significant kind of improvement in the device design and kind of just oxygen production that's really kind of helping these cells. And so we made improvements to the electronics encapsulation. So kind of ways to prevent that water ingress from biofouling these electrodes and kind of like almost like short circuiting kind of the bioelectronics, and then kind of looking at more mechanically reinforcing some of these gas and cell chambers to kind of prevent any I guess immune cell infiltration that we had some issues with the nanoporous membrane. So then we basically repeated that exact experiment, so loading 850 rat islets and transplanting into B6 mice. And now we can achieve kind of glycemic control up to 3 months, where once again, once we remove the device, we see a return to a hyperglycemic state in the animal. And so then we also measured the HbA1c as a kind of indicator of that glycemic control over a 3-month span. And so we do see, it's slightly elevated compared to a naive animal, but significantly reduced compared to a diabetic control animal. And that could be, still getting some of the, from the original FTZ kind of diabetic state, kind of spilling over since it's, for over 3 months. But then when we look at the glycated serum protein, which is more of an indicator of glycemic control for the past about 30 days, we don't see really any significant difference between kind of a naive animal. Once we remove the device, we see that they're viable. We performed another GSIS, so incubating under a low glucose and a high glucose solution and measuring the amount of insulin the cells are secreting. And so we do still see a modest increase in insulin secretion under high glucose, but it's not quite as good as when we retrieve the device at just 1 month. And so this is kind of one thing that we are kind of working on, is how can we kind of improve the insulin kinetics and improve kind of the, I'd say, happiness of the cells within our device. So then next, we wanted to see, these, human stem cell derived islets are, a really hot topic. Vertex published a really nice paper recently showing, their efficacy in human patients after intraportal naked islet transplant under immune suppression. So right in the future, we'd love to kind of incorporate these cells into our devices and see if we can get their survival without any immune suppression. So we loaded 12 million SC-derived beta cells into the device, and then once again did the subcutaneous implant into B6 mice. And so these cells were provided by the Millman lab at WashU in St. Louis. And so we see kind of a similar reduction in the hyperglycemia in these mice. However, we don't go to the same kind of below or hovering, around like 200 as we saw with the kind of primary rat islets. And so these cells, they do take some time to mature, about like 3 to 6 months. And then just the difference in, say, like insulin from a human, it's not as like efficient as compared to, say, like a rodent insulin. But so we are able to see, human C-peptide in the serum up to 9 weeks in these mice, and then a similar reduction in the, A1C. Now once again, we see that they're viable once we retrieve the device. And then they also stain positive for diphthozone. So diphthozone is kind of a red stain that stains mature insulin granules. So you can use that kind of as um, a method to, how well, how are these cells like really producing kind of insulin at a high capacity. So we did kind of observe that underneath this, PDMS support structure, where it's probably likely, not really getting very much oxygen. We also have kind of a low kind of cell viability kind of in that region. But kind of other areas where we, hypothesize are getting like adequate kind of diffusion of oxygen from our gas diffusion chamber, we do see nice C-peptide and glucagon staining. And kind of interesting, we also observed that glucagon, the alpha cells, seem to be more kind of within the core of the clusters versus kind of C-peptide and beta cells more on kind of the exterior of the clusters. So this could be kind of just, I don't know, we're still looking into it, but it could be kind of an influence of kind of the maturation process. And so, we're interested in how this is being affected kind of within kind of our device and in a subcutaneous kind of transplant location. So then we also did another kind of GSIS, and kind of similarly, we do see positive insulin secretion, but only a modest, I'd say, stimulation index, but definitely better than when we don't provide any oxygen for these cells. So then next, like mice are really great, but ideally we want to kind of see how this will function in a large animal that's closer to kind of a human patient. So we took rat islets, and now we kind of created a duplex device, so kind of two mouse devices hinged together with kind of a PDMS frame. Um, and so this is kind of what it looks like before we transplant, and then kind of right after the day of surgery. And this once again, it's really well tolerated by the animal. So this is after kind of 90 days, the hair grows back over the device, and the animals are relatively happy. So these are the glycemic controls. So you can once again see that we can return the animals to a normal glycemic state, and then return to a hyperglycemic state after we remove the devices around like either day 90 or day 100. And so once again, we see that they are viable. They don't have any, don't see a significant decrease or a significant difference in the glycated serum protein between a naive animal, suggesting that the past 30 days has been, blood glucose control has been like well maintained. And then these are also, glucose responsive and demonstrate actually the best of our kind of models that for a stimulation index compared to some of the other kind of transplant models that we performed in these studies. So, all right, rodents are great. Mice, we want to first be able to cure, a mouse and a rat, but ideally we want to translate this to large animals, and especially models that will be more kind of representative and more similar to kind of human patient. And so but for these, experiments, we could not use the same Neurolux cage, so the same kind of power harvesting scheme where the cage acts as the kind of the NFC zone to how the device basically harvests its power. So we designed basically a wearable system for the monkeys that could then power these devices. So we have a rechargeable battery. Then this goes to kind of a power management, a microcontroller that can then tune kind of the frequency to match the implant, basically the secondary implant or secondary antenna on the implant. And so this is kind of what all the kind of components look like. So here's a rechargeable battery, here's the kind of the NFC front end, and then there's this SMA cable that attaches to this flexible antenna that the matching, like network frequency matching, that then allows the device to harvest power. And so we also, had to design these kind of custom monkey jackets, which could hold these components, which then there's a wire basically inside this kind of backpack that feeds the to this kind of antenna that's placed on top of the implant. Um, and then, we do a lot of, wrapping with like gauze and different things to make sure that the animal doesn't, try to rip the this like antenna kind of off, which we went through a couple like iterations and had to kind of get the animal used to kind of wearing some of these systems before we did the actual implant study. And then here you can see we just have kind of an LED, so it's same, just to confirm that all of the electronics are working and that we are kind of generating that we are getting kind of power harvesting by the device. And so for this first study, we transplanted 3 oxygen devices subcutaneously. We also implanted in another location 1 control device, so without any oxygen. And actually, these were all on the back. It looks like it was on the stomach, but it was actually on the back of the primate. And we loaded about 12,000 IAQ per device, and then looked at kind of similar metrics, so the like viability, and some imaging stains. So we could show, so we retrieved these after 1 month, and we can see nice viability of the cells within the devices. So these are averages of all 3 devices, but all 3 were very similar, and we're able to maintain kind of a nice viability. And we use kind of 75% as a kind of biological reference value, since for naked islets, preparation has to be above kind of that threshold, above 75%, to be deemed suitable to go into a patient with type 1 diabetes. So we did see positive diphthozone staining. It wasn't quite as strong as we would have liked, so not quite as dense, indicating that there are insulin granules, but maybe not at the level that we would ideally want. But we did see measure the total insulin content per device, and we do see that they're able to produce significantly more insulin compared to if we don't provide any oxygen. So then, looking at kind of histology, so what's like the morphology of these cells? So with the oxygen devices, we do see nice kind of compact islet morphology on H&E staining. They also are producing C-peptide and glucagon, whereas they are control devices. All of those islets have lost their morphology. Many they've almost kind of disintegrated into these kind of either super tiny cell clusters or single cells. So in summary, we've shown that wirelessly powered oxygen generating macro device can enable glycemic control in FPV diabetic rodents for 90 days, so for up to 3 months in rodents. And we also protected viable allogeneic islet cells in a non-human primate model for 30 days. And so kind of in some of the future direction, we're always looking to kind of improve the durability of the implantable electronics. So kind of, preventing that biofouling, water ingress that can incapacitate kind of electronics. And then kind of more so focusing on improving the insulin kinetics with a sub-Q transplanted macro device. And so all of these devices, they did not have any anti-fibrotic coating. So there is an intact kind of fibrotic encapsulation layer, which can really just really hinder kind of exchange of, say, like other nutrients and like glucose, and kind of removal of waste products. So we're hoping that we can now begin to kind of apply these anti-fibrotic coatings, that we can kind of decrease that kind of barrier and improve kind of some of these insulin kinetics. But it has been a challenge. So some of the the chemistries that we've optimized for a lot of these either small molecule antifibrotic coatings or crystallized drug kind of formulations are there. They require organic solvents, and unfortunately, the proton exchange membrane is very kind of susceptible to any organic solvent. So we've been designing new chemistries, aqueous-based chemistries that hopefully we can kind of then apply some of these small molecules that we've discovered we can really modulate the immune system and put them towards a phenotype that is really conducive towards graft survival. And then we're also looking into improving potential, patient compliance and safety by incorporating, an implantable rechargeable battery. So kind of using our same antenna system, but now it would power basically to recharge a battery implant. And so kind of with this system, then we would be able to kind of incorporate some like alert systems for the patients in case, say, like oxygen levels were getting too low or battery life was, too low. And so kind of using the same either NFC coupling. We can also like monitor how the device is functioning so we can get kind of current measurements to kind of back calculate how much oxygen is being produced. And yeah, just I guess after kind of our monkey experiments, there was a challenge to kind of keep everything kind of situated on the private and prevent them from, kind of like tugging and pulling at wires. So we think, it may be more conducive to, patient compliance if we have kind of some sort of a rechargeable battery system. Um, and then, ideally, we would love to create like a closed loop system where we can simultaneously monitor the oxygen tension within the device, and then really only turn on oxygen generation when it's needed, and we can really fine tune kind of and optimize the delivery of oxygen to like the right like exact kind of like level that it's needed to promote graft function. And so, yeah, with that, I'd like to thank Dan and Bob for their excellent mentorship, especially Breakthrough T1D for their support of this project, Jeff Millman and Nathaniel for providing the SC islets, and then our collaborators at CellTrans doing all the monkey work, Jose Oberholzer, Peter Rios, and Daisy Lopez. But yeah, with that, I'd love to take any questions.
Thank you, Matt. This was really great. I wondered, just can you speak a little? I mean, two of the biggest things are, how can this device persist in context of, an upregulated autoimmune attack after it's implanted? And then, how do you easily monitor any changes in cell architecture or, um, any kinds of tumor genesis and be able to retrieve that device? I guess those are two biggies. You kind of touched on those, that those are issues, but how are you, thinking forward into solving those big issues?
Um, so let's see. So on like the retrievability of the device, that's what I think like is probably one of the best aspects kind of this device is that it's
Mhm.
super easy to retrieve just from a subcutaneous, like just underneath the skin, to easily be removed. But kind of like also the subcutaneous site is just it's very difficult. So right, there's just not really good mass exchange kind of other like nutrients. So oxygen availability is one is going to be super low, but there could be other nutrients that are required by the cells and like removal of waste products. So I
Mhm.
think we've found that just the subcutaneous environment is like really challenging for these macroencapsulated islets, especially when you move to, say, a large animal like a non-human primate that has a more, highly developed immune system, and just that level, that thickness of fibrosis is just like, I don't know, 10 times, 5 times is like
Yeah.
like as
Yeah.
compared to like say like a mouse. And so yeah, this has kind of been our challenge and like we debate. So I think the IP cavity has a lot of benefits in terms of mass transport of other, say, um, metabolites and other kind of like nutrients. So you get really kind of like circulation of IP fluid, and so that kind of like
Yeah.
washes the devices. So I think that really enhances kind of one the insulin kinetics and the exchange. But then, right, you have to have like the more invasive kind of surgery. So is that going to be adopted by like patients? So I think they're right. There's trade-offs, and so um, right. I mean, I'm not sure which is the best option. I think we're trying to still go after the subcutaneous site. So, like, can we kind of either remove this membrane? Can we have so we also use alginate, um, just as kind of more of a matrix to kind of hold the islet cells. But then that, in itself is it's it prevents any T cells and other cells from getting in. So like we're kind of almost creating this double barrier, right, by having this our PCT membrane. So, maybe we don't need that. But then we kind of have to look at kind of mechanical, like an influence on like mechanics and how do we kind of integrate, these two materials with say like different elastic modulus, one really stiff, one kind of more a hydrogel, and how do we safely kind of incorporate kind of these materials together. So yeah, these are like things that
What's
we're
the
thinking of.
best model system currently in other disease states for this implantable secreting, unit? Not even
Um,
like taking away the autoimmune issue.
so so we've applied this device with an engineered cell line, so with ARPE cells that
Mhm.
are engineered to secrete biologics. So kind of antibody based drugs that can either as like inhibitors for can like prevent I don't know like PD-L1 or CTLA4 um kind of so kind of more for say like a cancer model.
Mhm.
um and so kind of I guess what's kind of nice with those applications is that you don't need as high of a cell density as compared to with islets. um and then they don't seem to require quite as much oxygen. And also islets are just like oxygen guzzlers. So I think that that's kind of the model that we've also been exploring, as a potential application for this device.
Yeah. All right. Well, we're hoping that the Oberhauser team, along with the Anderson and Langer groups, can push this forward a bit, and it would be great to see it getting smaller, faster, and better. Yeah,
Yeah, exactly. Yeah, we're trying to do all those things. So
great. We'll keep going. Thank you
yeah,
so much for chatting with us today. I really appreciate it, and have a good rest of your day and a good Labor Day weekend.
yeah. Thank you. You as well.
Thank
And
you.
thanks. Yeah, thanks so much for inviting me.
Thanks, man. Thanks for
Yeah,
sharing. Bye-bye.
yeah.